Hello everyone, thank you for being here. Today we are going to dive into the fascinating world of microbiology and explore the essential techniques that allow us to study these microscopic organisms. To start, let's define microbiology. It's a field dedicated to studying microorganisms, tiny living things that are invisible to us. This includes well-known microbes like bacteria and viruses, and also fungi, algae, and protozoa. It's a common misconception that all microbes are bad. In reality, while some can cause diseases, most are incredibly important for our planet's ecosystems, our food supply, and even our own health. The journey of microbiology began in the 17th century with a pivotal innovation, the microscope. A Dutch scientist named Anton van Leeuwenhoek created a microscope powerful enough to see things no one had ever seen before. He was the first person to describe these tiny living creatures, which he called animalcules. His incredible work is why he is known as the father of microbiology. The late 19th century is often called the golden era of microbiology. It's when the field truly became a formal science. This era was defined by two scientists, Louis Pasteur and Robert Koch. Pasteur proved that microbes don't just appear out of nowhere, and his work led to the pasteurization process we still use today to make our food safe. Robert Koch, on the other hand, developed a method to scientifically link a specific microbe to a specific disease, which was crucial for the development of modern medical microbiology. Moving into the 20th century, the discovery of penicillin by Alexander Fleming launched the age of antibiotics, marking another major turning point. Today, we are in the second golden age with advanced tools like DNA sequencing, helping us understand the human microbiome and find new ways to use microbes for everything from medicine to environmental cleanup. The field of microbiology is vast, so it's broken down into sub-disciplines. The first main branch is pure microbiology, which is the fundamental basic study of these microorganisms. The second branch is applied microbiology, where we use the knowledge in practical ways. I have listed a few examples here like medical microbiology for studying diseases and industrial microbiology for things like making food and pharmaceuticals. So, what are microbiological techniques? Simply put, they are the tools and methods we use to work with microbes in a safe, controlled way. These techniques allow us to do things like identify a specific bacterium, grow it in the lab, and study its properties. The most crucial principle here is aseptic technique. This means working in a way that prevents contamination from unwanted microbes, which is the foundation of all good microbiological work. The fundamental techniques of microbiology can be remembered with the 5 I's. Let's walk through them. First, we have inoculation, which is simply taking a sample and placing it into a medium where it can grow. The next, we incubate it, providing the right temperature and conditions for the microbes to multiply. Once they have grown, we perform isolation to get a pure culture of a single species. Then we inspect the culture and the cells under a microscope. And finally, we perform various tests for identification to figure out exactly what microbes we are looking at. Let's break down some specific techniques, starting with aseptic and culturing techniques. As I mentioned, aseptic techniques are all about preventing contaminations. This involves practice like sterilizing our equipments and disinfecting our work surface. We use different methods like heat or radiation for sterilization, which completely eliminates all microbes. For surface, we use disinfections, which kills most but not necessarily all microbes. After ensuring a sterile environment, we can use culturing techniques to grow our microbes. We use different types of media. Liquid media is for growing large volume, while solid media like the agar plate you see in labs is used for isolating individual colonies. For isolation, the streak plate method is the most common, where we spread the sample across the agar plate to get single colonies. The pure plate method is used to count the number of microbes in a sample. Microbes are often transparent, so we use staining techniques to make them visible under a microscope. A simple stain uses just one dye to color the cells, which help us see their basic shape and arrangement. A more powerful technique is differential staining, like the famous Gram stain, which uses multiple dyes to distinguish between different types of bacteria based on their cell wall structure. There are different types of microscopes we use. The light microscope is the standard tool in most labs, allowing us to see stained or living microbes. For much higher magnification, we use an electron microscope. This allows us to see the tiny details within a cell and even the structures of viruses. Beyond observation, we can use biochemical and molecular techniques to identify microbes. Biochemical tests analyze what a microbe consumes or produces, like fragmentation test. Molecular techniques, on the other hand, are much more precise and look at the microbe's genetic code. Techniques like PCR can amplify tiny amount of DNA to detect a pathogen, and DNA sequencing can give us the exact genetic blueprint for a microbe, providing the most accurate identification. Immunological techniques use the body's natural defense system to identify microbes by looking for how antibodies react with them. This is how we can diagnose diseases. And finally, we have antimicrobial sensitive testing. This is a crucial technique for medical microbiology as it tells us which antibiotic will work best to treat a specific infection. The Care by Bayer method is a classic way to do this, where we look for a clear area around an antibiotic disc called the zone of inhibition, which shows that the antibiotic is effective. So, why are all these techniques so important? They have a huge impact on our daily lives. They are essential in disease diagnosis and help doctors choose the right treatment. They keep our food and beverages safe and allow us to make things like cheese and yogurt. In the pharmaceutical industry, they are vital for quality control and producing life-saving medications. And they have a major role in environmental and agricultural applications, from cleaning up oil spills to studying beneficial soil microbes. Before we conclude, it is crucial to talk about safety. Working with microbes requires strict laboratory safety and precautions. This includes wearing the right personal protective equipment like a lab coat, glasses, and gloves. We must also consistently disinfect our work area and use proper aseptic handling techniques. Proper waste disposal is non-negotiable, with all contaminated materials going into biohazard bags. The most important rule is to treat all cultures as potential biohazards and handle them with extreme care to protect ourselves and others. In summary, microbiological techniques are the fundamental tools that have allowed us to understand and interact with the invisible world of microbes, from the first observation by Van Leeuwenhoek to the advanced molecular methods of today. They are vital for medicine, industry, and our environment. Thank you for your attention.